It could be a lack of red blood cells. That's correct. That's one way of getting anemia. So the word is, let me write it down. Anemia. What does that mean? Yeah. It could be iron. It could be red blood cells. Not white. Well, the B12 led to something. So the question is, what is anemia? And in general, anemia is a lack of ability to get oxygen to the cells. So you can't get oxygen to your cells, so you can't do cellular respiration very well, so you can't make ATP very well. What do you think is going to be the result of that?
Maybe could be like death.
Could end up dying. Yeah, my buddy died of this, but what is? Yeah. That's right. Lack of energy. You'll hear people saying they're cold. You'll hear people say they're fatigued or tired. Easily tired. Yeah, they might be associated, but unlikely. I don't know. Asthma has to do with the lungs and breathing. Asthma could lead to some of this if you're having trouble breathing. I guess. But this is more, you're just living your normal life, and you're just tired. You're out of breath easily. That could be a link to asthma, but not. Right? So your lack of energy, you're cold, you're tired, you're out of breath. Doctors are going to start thinking, well, let's check and see what's going on. And how do you get oxygen from your lungs to your cells? You have to use red blood cells. And red blood cells have in their center, a mature red blood cell got rid of their nucleus, got rid of almost every organelle. The only thing in a mature red blood cell are these 4 chains. And I'm just going to draw them simply. 2 chains, 2 alphas. There's still talking going on. It's really becoming to the point where it's I don't know what to say. It's really interrupting class. So stop it. There's 2 alphas and 2 betas. Now, we got to understand that these are proteins that together they're polypeptides. They're made by genes. So there's a gene that makes the betas, and there's a gene that makes the alpha. And together they come together and form like a cradle, a nest, where an iron atom sits, an iron ion sits in a center, ready to capture oxygen. Is that clear? That's how this works normally. Iron captures the oxygen. See, it has to. If you allow oxygen to associate with iron ion, what's going to happen is the iron's going to rust. But that means it's going to bond tightly. We don't want the oxygen to bond tightly, do we? We want it to bond enough for the red blood cell to carry it to the cells, and then we want those red blood cells to let the oxygen go. Because otherwise, the oxygen's stuck. We don't need it stuck. So the red blood, that's just an outline. Obviously, this doesn't actually look this way. So that's what we're dealing with. 2 genes. Now in the case, they got this letter. Then can you even read it?
It's on the next page.
Oh, okay. So the letter reads as so. Dear Dr. Castle, I'm writing you in the hopes that securing a research position in your lab I'm completing my last year of medical school at Johns Hopkins and would be available to move in August. 2 years ago, I saw my first case of sickle cell anemia. I was so intrigued by the case that I decided to see if I could investigate more of the physiological mechanisms that led to the symptoms these individuals display. Recently, I compared the transmission spectrum of blood obtained from a normal patient with that of a patient with sickle cell anemia. My data indicate that there is a difference in the wavelengths of light are optimally transmitted through the blood of sickle cell individuals. I read through your journal article in which you report that sickle cells flow through the cardiovascular system at the slower velocity than normal shaped red blood cells. That I do hope that you still have an interest in sickle cell anemia. I feel that I would make a positive addition to your lab. I am most interested in being able to investigate whether this difference in light transmission could account for the physiological complications seen in this disease. So, to be clear, a normal red blood cell looked at from the side looks something like this. It's rounded. It fits through the capillaries easily. The hemoglobin's inside. It only lives 3 months because it's gotten rid of all its DNA. So it only can last 3 months. So every 3 months you have to replace your red blood cells. A sickled cell looks something like this. It moves slower through the bloodstream because it has pointy ends. It's odd shape, gets stuck in the capillaries often. If it gets stuck for a while and you forms a clot, that could cause what's called a sickle cell crisis. All right? So that's a blood clot that will stop flow of blood to that region, whether it's small, it could be in a toe or in a finger, or it could be in an eye or a liver or heart. If it happens in the eye, you could go blind. If it happens in your heart, you could have a heart attack in your brain, and we call it a stroke. Yeah.
How does this protect you from malaria?
That's a good question. Let's talk about that at the end of this. But it's a good question. Keep that question in mind.
You said that if it gets stuck in the heart, it can cause you to go blind.
If the blood clot happens in a major artery that feeds your eye, then it'll kill your eye. Any blood clot is damaging the tissues, because a blood clot means you're stopping the flow of oxygen. The cells are dying. You have a limited amount of time to get oxygen to those cells. Minutes. Right? And you have to do it carefully. There's all kinds of physiological discussions you could have on how you do that. It could mean surgery. It's unlikely. It's probably going to mean a medicine that's injected into your veins. Yes. Yes, but blood clots are normal. What's abnormal is having blood clots in places where you're not actively bleeding. So you should be having blood clots if you're bleeding. That blood clots are what stop you from bleeding out. Does that make sense? So during your period, to answer your question, you do get blood clots. That's what comes out of, you don't usually get just a flow of blood, correct? It's clotted blood. That's normal. Platelets have formed clots in order to stop the bleeding. The blood is actually coming from the extra uterine tissue that had developed and waiting for implantation of the egg. Yeah.
So is major loss of blood just an oxygen issue?
I guess you could look at it that way. Yeah. It's the reason everything else dies is the blood is carrying oxygen. Of course, it's also carrying sugar and nutrients, but the sugar and nutrients are secondary to oxygen. Without oxygen, the mitochondria, the mitochondria, it does not work without oxygen. There's no final electron acceptor. The system stops. No ATP is made. Yeah.
Do these cells have the same life expectancy?
Same 3 months. It's a good question. I do not know the answer to. I would guess yes. I would guess it's still 3 months, but it could be shorter, it could be longer. Good question. Yes. So before we go on, let's talk about this idea of hemoglobin. let's remember it's a polypeptide. Do you remember that word? Okay, good. Polypeptide. And that means that, remember, we had a basic amino acid, that NH2, it had a center carbon, had a hydrogen on one side, and some kind of R group on this side, and then it was connected to another carbon, C double bond O, OH. You remember this? All right, that was an amino acid. And we said there were 21 different amino acids because there were 21 R groups, or something we called side chains. And we said that R groups or side chains could be nonpolar. Some of them might be nonpolar. Some might be polar. Some might be large. Some might be small. Some might be very positively charged. Some might be very negative. So so if you have this big long chain called, let's look at just this example of hemoglobin. Just looking at this one hemoglobin, let's say alpha, the alpha chain. If that means that this is a big long chain of amino acids, one bonded by peptide bond in the next, all the way down hundreds, right? Hundreds of amino acids. What if most of these were negative? What would the protein be? What would this protein be?
Polypeptide.
It's a polypeptide, but what would this polypeptide be? If negative, because most of the amino acids are negative. If most of these amino acids were positive, what would this polypeptide be? Positive. If most of these amino acids were acidic, what would this polypeptide be? Acidic, right? So the properties of the polypeptide are determined by the amino acid. Do you agree? Everybody agree with that? The properties of the polypeptide of this protein are determined by its components, its amino acids. Do you see that? Is there confusion about that? So hundreds of amino acids, it's what the amino acids are and what their order is determines the chemical function, the structure, and the chemical function of this hemoglobin. Does that make sense? So if I just change even one amino acid, if I just change one, then I could have a change in the chemistry of this polypeptide. Now the polypeptide is something different. Now maybe it's not as positive, or maybe it's more positive, or maybe it's not as negative. I don't know. But whatever change happened, it happened because of this, because I changed an amino acid. Is that understood? How do you know when you make hemoglobin, how do you know what amino acid goes where? Made in the cell, correct? How does your cell know what amino acid to put when? How does it know what order of which of the 21 amino acids to put together to make hemoglobin? Where's the recipe for this protein? This protein is in DNA. It's in the thing called a gene. It's in a gene. And a gene, as you should know by now, without any question, should be no question at this point, A gene is a region of the DNA that codes for a protein. The question was, how does the cell know to make hemoglobin versus skin? How does the cell know that it needs these amino acids in this order? By the way, we call that the primary structure. It's the order of the amino acids. Is how does it know what the order of the amino acid is? It's the gene. So if you inherit the big S, you get the normal hemoglobin. You get the little s, you got, and I'm just going to put big S, little s with a prime so you know it's little. The little s, you get two little s's, and you have the sickle cell hemoglobin. Is that clear? In fact, just one little s gives you the sickle cell hemoglobin. If you have two little s's, you're making only sickle cell hemoglobin, so that means you have the disease. So it's recessive. And it's all based on this idea of genes producing a protein, deciding the order of amino acid. Go right to it. So in this problem, they purified hemoglobin from blood samples and sent the subject through electrophoresis. Now let's talk about that. What is electrophoresis? You should know what it is. Good guess. So, yeah.
Can I get another one? Because this packet is missing pages.
So you put the material in a gel. Do you remember when we did osmosis and diffusion?
Yeah.
Remember that gel we cut out? Well, we're going to use it again. We have some machines. These machines, we have them. My objective is to have you do this. All right? So these gels are going to have, I can have another, if there's still issues, I can make more copies later. Right now I need to focus on this, okay? They have these things that we call wells. They're called wells. And wells, this is all in water. It's in a solution that conducts electricity. So it's actually a buffer. So it keeps the pH at a constant. So this is all happening. This whole machine is connected to wires. There's several ways you can connect them. Several different machines. There's horizontal ones and vertical ones, big ones and small ones, etc. We connect it to electricity, usually a battery, sometimes a power source. And you take a protein like during the lab, but what they did here is they took the hemoglobin from the different people. Please note that they tell you in the problem they had 5 patients. And they took the hemoglobin from 5 different patients. 1, 2, 3, 4, and 5. They had different ID numbers. These are the ID numbers. They took these samples, they took the hemoglobin, and they put it in the well. Okay, that's the blue there. And they ran the electricity, so they plugged it in. Now these are negatively charged. What do you think is going to happen to the protein?
It's going to be negative.
No, the protein's negative. If you run electricity through that? What does negative do to no? You have a negative charge, a negative electrical charge here. These have negative charges. The proteins have negative charges. What's going to happen? They're floating in water. This is a gel.
They're going to attract to the positive.
They're going to attract to the positive, so they're going to move in which direction? Down. Now, somebody said it. I'm glad. I didn't hear it, though. So they're going to move downward. they're going to move downward in two different ways to separate. There's at least two different ways of separating things with gel electrophoresis. The way they do it here, so let's call that number one. The way they're using it here is they're going to separate. You're going to separate by charge. So the more charge, the better the separation. Is that clear? So that means, is it going to move faster or slower through the gel? Faster. Good. The second way, which we're not going to do here, but we'll do in the lab that I'm planning, is that we will separate by size. So the bigger the molecule, the slower it moves. The smaller the molecule, the faster it moves. Okay. So, but in this problem, what they chose is to look at what the charge of the amino acid was. Was the amino acid highly positive or highly negative? I'll have to wait. Okay, go ahead. So there's two ways gel electrophoresis can separate molecules. Okay, so if you want to read this, you have to turn it like this because here's the origin. This is where they started. To read it, you have to understand that they put the hemoglobin here at the beginning. So this is where they began. All the hemoglobin was put in wells. And if you were looking at this, the well would look something like this. You'd put the hemoglobin was put in these wells. Is that understood? You turn on the electricity. Of course, you use something that's going to bind to the protein relatively specifically.
What I mean by relatively specifically is that when you isolate and purify a protein, you have this protein that's sitting there in a beaker, but it's also, there's a lot of other proteins that are there. Some of them might be similar. We've isolated it, you purified it. As you can see, so we purified the hemoglobin. And so we already identified that there's two, when we're talking about hemoglobin, even though you purified it, that's a kind of a tricky word because there are levels of purification, right? It's hard to get something 100% pure. So when we use dyes, what usually happens is that you have some type of specific binding agent. Sometimes it's antibodies, sometimes it's another piece of protein. An antibody can be is a protein. It might be some other molecule that's attached to a molecule that will absorb light. That is your pigment, right? It's a pigment molecule, sort of like melanin is for our skin, and so it attaches. Normally, they attach specifically to what we want. So let's say it attaches to hemoglobin. The white strand here is the hemoglobin protein. So it's going to attach. It's going to be directed towards attaching specifically to that protein. So when you look at the gel, the only thing you're going to see is what this pigment is attached to, and that should be only hemoglobin. All right, so there you have it. That's the idea. And so you use this electrophoresis gel. It's a gel, so you have to consider when you're looking at a gel that it acts as a matrix. So you have these strands of fibers. That's what a gel is. These polymers that act to hold on to each other like little strips of Velcro. They hold on to each other, and in between them is all this buffer. We call it a buffer, right? So there's a liquid, and that liquid is a buffer. And what all that means is that it's going to keep a constant pH. constant pH or acidity, right? It's either going to be acid, base, neutral, whatever you set it at. And what's interesting is that the ones that are more, you can, again, it could be you're separating it by size, or it could be you're separating it by charge. And the details of how that's done is not really pertinent to this discussion. But when you're looking at this and you're deciding, well, how do we separate these? Well, they decided in this example to separate these proteins uh, in this column by how charged they are. So you put the protein here in the column, and as you turn on the electricity, as someone mentioned, the protein starts to move its way through this matrix. The more charged this is, the faster it moves through this matrix. So the further down it goes, the less charged it is, the less negative it is, the slower it moves through this matrix, right? So you have these columns. There, as you can see here, there's this column here as the protein moves through the gel. This is a side view of this gel. This is the front view. This is the side view. So you put the protein at the top in the well. In each one, you put a different sample. You have sample 5 here. You have sample 4 here, right? You have sample 3 here. You have sample 2 here. And you have sample 1 here, right? Well, it kind of is a little bit off, but let's just put it here. So then what you'll see is that as you run the gel, we say running the gel, meaning that the proteins are running through the matrix, they start to separate. I'm sorry, there's a faculty meeting and the people in the hallway talking. You see they're as loud as you guys are. So as you see, they separate from each other in comparison to one another. They're in different lanes, so they run down. They only run down. And so the question is, 2 of these samples have sickle cell, and 2 of these samples don't have sickle cell. Or I'm sorry, 3 of the samples don't, and 2 samples of this hemoglobin are from sickle cell patients. So the question is there a difference? And remember, in this case, they don't know what causes sickle cell. So they're asking, is there a difference in the hemoglobin? So the doctor that the letter was sent to says, oh yeah, I can see that there's a difference in the patients. Could you let me know who had sickle cell and who didn't? And there's a question that they ask on the next page is, why would the doctors not tell them who had sickle cell, who didn't? And one of the other students and one of your colleagues in the class mentioned that the reason that they did that is because they didn't want to bias the professor with the results, by helping them interpret the results. They wanted just to give them the pure results. These are the samples. Do you see what I see, kind of thing? And he said, yeah, I see it. Which one of these has sickle cell, which doesn't? So 2 of them have sickle cell. I'm going to pause for a second. Can you identify which 2 have the sickle cell and which 3 don't? So when we look at these results, we see that two columns are very much alike. This column 5 and this column 2 are very much alike. They're in the same place in the gel. So these 2 columns have the same charge. Have the same charge. This has a hemoglobin that's the same charge, and these 3 have a hemoglobin that have the same that are different. So these 2 individuals have sickle cell disease. These 3 do not have sickle cell disease. So let's pause here and think about this. When you're looking at
a gel. You're looking at the differences between the substances that you put in the wells. And again, depending on how the gel separates the material, in this case, it's separating these particles, these molecules, not by size, but by charge. The more negative the substance is, the more negative the substance is, the further down it goes, down this column, this gel. So these three here are very negative. These two are less negative. So is this one here. Now, assuming that there's no difference in the affinity of the dye, the specificity of the dye, in other words, assuming that there's no error, there are two molecules here, both of them hemoglobin, one that's the same as these two, and one that's the same as these two. So in other words, this is the normal hemoglobin here, and we know that because we know what the samples were. These two individuals, this person had the disease. So let's be clear. We know from the physician, from the letter, that number 2 and number 5 have the disease. Number 2 and number 5. Remember, they were separated by this in this column. And 2 and 5 have the same charge. Now remember what we talked about earlier, that means that they have the same, similar, at least similar, in this case we know because of the experimental design, the same amino acid sequence. Right? Amino acid 1, amino acid 2. So they're equally, all the amino acids are equally less positive here, where these are equally negative or more negative. It looks like all the hemoglobin in this person is the same, and they're not diseased. They're normal hemoglobin, so this would be normal hemoglobin. This person has all the hemoglobin is the same, and that's all normal hemoglobin. This person has all the hemoglobin is the same, and it's abnormal hemoglobin. So again, sickle cell. And we know from the discussion that that's because this hemoglobin under low oxygen concentrations sickles the cell. Remember, that means the cell looks like this, right? Something like that. So this person has a disease. This person has a disease. All the same hemoglobin. All the hemoglobin is changed. Has that odd amino acid that's different from the normal hemoglobin that makes it less negative. Okay. So this is what the gel confirms for us, is that the hemoglobin is different here. 2 and 5 show us that the hemoglobin is different. And it's different at the amino acid sequence. So that means the gene is different. Because remember, a gene leads to an amino acid sequence. Creates a peptide, a polypeptide, which we covered already in the first semester. So the gene produces the polypeptide. So we know that there's a gene that produces this abnormal hemoglobin for 2 and 5. They had the disease. These people are not diseased, and all their hemoglobin is the same. So 1 and 3 have the same genes. So to be clear, 1 and 3 have the same genes. the same genes, only one kind of protein is made. How do I know only one kind of protein is made? There's only one band. You see here? 1 and 3 only have one band. In these two people, they have the disease, but they also only have one kind of hemoglobin, one gene. So they have 2 copies of the same gene. These people have 2 copies of the same gene. So these have the normal gene. 1 and 3 have the normal gene. That equals the normal hemoglobin. By the way, that's my dog snoring in the background. Sorry about that. Where 2 and 5 have the abnormal gene, both 2 copies, by the way, 2 times. How do I know there's 2 times? Because if it only had 1 copy, if it only had, for instance, we know that this is the dominant allele, so this has 2 S's, 2 big S's, right? How do I know they have 2 big S's? Because you only have 1 band in 1 and 3 only have 1 band, so they're only producing 1 kind of protein. That means they have the same genes and they're normal, and normal is dominant. So we know that, well, we don't even know it's dominant yet, but we will by the end of this discussion. These two have only, they're normal. They have only 1 band, so there's only 1 gene, so they only have 1 type of gene, 1 allele. But we know they have 2 copies of the gene, so they have double dominant S's. 2 and 5 only have 1 copy of the gene, 1 version of the gene. You see there's only 1 protein that's being made. We know that because of where the band is. So if they're in the same area, if they're in the same horizontal line, then they're the same for all intents and purposes, to make it quick and short and dirty. If they're in these proteins all have similar qualities. In this case, they all have the same negativity. So this one and this 5 and 2 and 5 are both abnormal. They're both sickle cell. So they're little s. And why don't we make sure we understand that and put a little hyphen there to make sure we know that's the capital. So then we know, how do we know that this is recessive? How do we know sickle cell is recessive? Besides looking at the environment, looking at people, looking at marriages, etc. We know that it's recessive because we can look at this person here, number 12. So we had some control groups. These 3 were our control groups. They were the people without the disease. And we're comparing the 2 people with the disease to the people without. But it's interesting, this person did not have the disease. So they're normal for the phenotype. Their phenotype is not sickle cell. Their cells are not sickled. But they do have a copy of the abnormal hemoglobin. They do have a copy of the abnormal hemoglobin. They have the copy of the normal hemoglobin, but they also have the copy of the abnormal hemoglobin. So how can we interpret that? What could possibly be the answer to that? There's 2 copies, so that means there's 2 different proteins. One hemoglobin is abnormal or sickle cell. And the other is normal hemoglobin. So I have 2 copies. They have version 1 and version 2. Again, you have 2 copies. There's 1 protein here that's abnormal, that's normal, and another version that's abnormal. So you have a normal, and an abnormal hemoglobin. So what does that mean? That means that they have two different alleles. Same gene, because both genes, the same gene controls hemoglobin. Only 1 gene controls hemoglobin, but there's 2 alleles. That means they have the sickle cell allele, which is in this case small s, and they have the normal hemoglobin allele, so they have the capital S. So number 4 in this problem, number 4 is the heterozygote. And they're normal. They don't have the disease. They are, when I mean normal, I mean average. They don't have the disease. They're non-diseased. Where the other two, which again we said 1 and 3, they have the normal, they're also not diseased, average hemoglobin. They must have the dominant form of both alleles because they only have one type of hemoglobin. It's not diseased. They don't get the sickle cell. This has 2 kinds of hemoglobin, one that's associated with disease and one that's associated with non-disease. So that means 2 different alleles, because that's how you get different proteins. You get different versions of the gene. I've said it like 5 different ways, all meaning the same thing, that a gene leads to a protein. When we look here at 2 and 5, 2 and 5 are both the same, but they're diseased. So we're going to label it little s, little s. So this is the homozygous recessive, which is sickle cell. That's associated with 2 and 5 is associated with the polypeptide that's diseased, that sickles the cell. That means it has a gene. And since there's only one band in the electrophoresis, only one band when we separate the hemoglobins, we know that it's the homozygous recessive. Then we can look at the normal gene or the average gene, the non-diseased gene, 1 and 3. Again, there's only one band. It's non-disease, so we know they don't sickle. Normal hemoglobin, average hemoglobin. We know 1 and 3 must be the dominant form. How do we know that normal hemoglobin is dominant? Not because it's not disease. You can have a disease that's dominant, but in this case, it's that when we look at the heterozygote, here, it's not diseased. Number 4 is not diseased, and it has 2 different versions of the hemoglobin, one that's sickled and one that doesn't lead to sickling, that's associated with non-sickled cells, what we call the normal. And which one shows up? The normal. So the normal must be the dominant form. So this is a really great case study. It allows us to kind of apply a lot of the things that we talked about with dominant and recessive. It allows you to kind of think about how things are separated, to look at this puzzle, and really it comes down to which one of these are the same and which are different, and then trying to figure out why this column, which is the non-disease, has 2 bands. So remember, when we separate with electrophoresis, what are our take-homes here? When we separate with electrophoresis, all we know is, we know 2 things. We know which, whatever they are, whether it's DNA, RNA, or protein, which molecules are similar and which are different. That's all you're looking at here. To understand what that means, how are they different, you have to understand the experiment. And they explained it here that the separation in this case was by charge, and I explained to you how that works in the gel. But the point is that they're different, and because the molecules in the same row are similar and the ones that are in different rows are different, please be careful and read the paragraph in a problem to figure out what is it that they're using to separate them. But the bottom line is you're separating them, so you get 2 different groups. You have the normal, and then you have the abnormal in this case, or you have the average, you have the non-average, or in this case, you have the diseased and non-diseased individuals. And of the non-diseased, of these three, this one is also different because it has two copies, two different proteins in the same column, suggesting because of how you get a protein from a gene, suggesting that these two proteins, that there's two different genes here, two different alleles, where all the rest of them only have one allele. So, to be fair, in a real gel, you do sometimes get what's called non-specific binding to the dye and all this other stuff. But it just comes down to what's similar, what's different, assuming there's no artifacts from or mistakes or errors in the results, which do crop up. Assuming these results are relatively error-free, then you can compare the control groups, the non-diseased individuals, 1, 2, and 3, to the diseased individuals, and then come up with some real understanding of what's going on. So gel electrophoresis separates molecules, in this case proteins, but it could separate RNA or DNA as well. Different electrophoreses are used for different conditions and different pHs, et cetera, et cetera. It separates by either size or charge. In this case, it was charge. Size or charge differences in proteins. Let's write that down. Let's just go ahead and write all this down. 2, electrophoresis separates molecules by size or charge, depending on how you set up the experiment. You have to read and think in order to figure out how the particular electrophoresis is working. They usually tell you in the problem. 3, now you know that they separate by charge. You know, in this case, that there's going to be differences. You know, you have control groups. You know, you have experimental groups. In this case, the experimental group was sickle cell group, the sickle cell patients, and the control groups were the non-sickle cell. So this was a true experiment. Which molecules were, So what electrophoresis helps you identify, or when you're looking at a gel, you're looking at which molecules are different, and you know that the electrophoresis separated them by either charge or size. The last thing you need to remember is that when you're talking about a protein, differences in charge or size mean differences in the gene. Okay? So when you break this down, if you see a difference here in these proteins, that means there's a difference in the gene. And if there's a difference in the gene, then you're probably talking about a different allele or a mutation, some kind of mutation. So that's how you get differences in genes, you get mutations. All right. So I hope this helped you with this case study. That was a great case study. I heard your discussions in class. They were fantastic. I know we can just keep developing that, and you will likely see these gel electrophoresis on your tests, on your standardized tests, but you certainly will see it in class. Didn't come and look at these results. Linus Pauling just sent to me. That was a joke. Linus Pauling's a famous chemist. So just so you know. He called, physicist too. He called the research assistant. He handed the letter to Jim and said, the samples, this and this, are from some patients who have been diagnosed with sickle cell anemia. The others are controls. So we have 2 48WCO3 and 48WC15. All right.
Sorry, does I have a question?
Yeah.
So for those that are less like charged, would that show like sickle cell or no?
And that was a really good point. And as you saw in the previous discussion, that's exactly right. It's not that the less charged shows sickle cell, is that we knew that the 2 individuals with sickle cell had hemoglobins that were less charged. Sickle cell is the phenotype. That's what we see. And then when we run our tests, we can compare what kinds of test results do you get when you have a patient with sickle cell. In this case, a protein that's less charged. A hemoglobin protein that's less charged. From the results, what level of protein structure in hemoglobin? Well, it's the primary structure, as we discussed earlier, when we're talking about the order of amino acids. We're talking about primary structure. Because it's the amino acid is either charged or it's not charged. So the order of those amino acids, which amino acids you put into the protein, is the primary structure. And primary structure is directly controlled from the gene. The gene controls the primary structure. Our Linus Pauling's results supported by Vernon's Ingram's results. Compare the molecular composition of the different amino acids implicated. And we just did all that work previously, right? Okay, let's move on. No, they knew that the people had sickle cell. By the way, there's your sickled cells. Do you see the difference? This is the normal cell here. This is what it looks like. And you can see that this, you would think that this would go through a capillary, a tube, easier than something like this, correct?
Yeah.
So you can see why someone with sickle cell would have
A lot of them are like weird. No, not all of them are that same shape.
Yeah, the shape is not, a lot of them have this sickle cell, but they might have all kinds of different shapes. The key is, and they might be in the middle of transitioning, because they go from this shape, they turn into this once they mature. So a lot of stuff is happening. These might be dying or dead. So they might be artifacts from the plating. I'm not sure. You guys read this. It's interesting. As you can see, mammals, right? Plasma cell, red blood cells in the plasma don't have a nucleus, right? There's the benefit, I think, if you're looking at the question, the benefit is that you have more room for hemoglobin. You're trying to, the whole point of that red blood cells is to carry oxygen. It's key. Predict how sickling of the red blood cells could impair function. Their function, we already talked about that. They get stuck. Predict how the average lifespan of a cell located in the brain differs from an average lifespan of red blood cell.
I'm sorry, so where are you getting this from? It's not here. None of these questions are on here.
It's on the version you were assigned on the day of. It's in your. You guys don't have this?
No, it's missing. Yeah, there's missing pages in this. Online may have all that.
The online version you had the day of the activity. You did have these questions, correct?
Yeah.
Okay, so I'm answering the questions that were there. This, obviously, something went wrong with these photocopies. All right? Were you not here the day of the activity?
No, I was here. This was the packet from the copies that were made.
Oh, so you had the packets first thing.
Yeah. But
And you guys used that instead of the online version and didn't have the questions?
I used the online.
Okay, so sorry, guys. All right, so let's move on. Let's just answer the questions. Is that all right? Predict the average lifespan of cell located in the brain differs from. Well, the brain is a life is a cell that lasts your whole life. Correct. It's the average lifespan of a brain cell is probably 70 some years. A red blood cell lasts 3 months, so you should know that. Basis. You would expect that because there's no nucleus in a red blood cell, so it can't survive long. Yeah, absence of organelles, it can't last that long. So 3 months is actually impressive. It has been observed using electron microscopes that when the red blood cells are sickled, there are little spikes in punctured plasma membrane. Predict how this will impact the function of sickle cell in their lifespan. Well, they're not going to last long with a membrane that's punctured. So who asked the question if they were longer? It's probably a shorter lifespan. Yeah.
Would that also affect the selective permeability instead of last?
Yes, absolutely. Selective permeability is impacted, and also physically it gets stuck. Are we all good with this?
Yeah.
How was the environment of the red of the blood different at the top of the tube versus the bottom of the tube? What does it say here? So again, it's the oxygen concentration impacts the shape of the tube. The higher the oxygen concentration, the more normal the cell. The less oxygen concentration, the more sickled it becomes. How would shaking the tube after the environment of the tube consider? If you shake the tube, the oxygen would be better distributed. What environmental factor do you believe is responsible for causing cells to sickle? The oxygen concentration. Yeah.
Would the concentration of oxygen in the air as well be affected in that?
Yeah, absolutely. In the lungs, could someone close that door, please? While the blood is near the lungs, the cell. I don't understand. When the blood cells are near the lungs, there's high oxygen concentration. The cells are unsickled. They look normal. But as your heart pumps that blood throughout the rest of your body, bless you, there's less and less oxygen. When you get down to your toe, this very little oxygen is compared to your lungs. That oxygen concentration in the environment sickles the cell. And that sickling and unsickling, filling up, and what happens? What do you do? What happens to a wire when you bend it back and forth over a period of time? It gets weakened. Plastic does it as well. And that's what's going to happen to this cell. As it keeps sickling and unsickling, sickling and unsickling, sooner or later it's going to burst. Its membrane integrity is going to be compromised, and it will leak, and it will die. And that answers your question that the sickle cell will have a shorter lifespan than a normal red blood cell. What environmental factor do you believe is responsible for causing? Okay, how would you repeat it? Okay, we answered those questions. All right, we're good.
Yeah.
So, if low oxygen is a factor, then causing the cells to sickle, actually, blah blah blah. Also triggered. Ghost interrupted Christina. So I want to answer your question because it's important. So here's an interesting thing here. Apparently he was able to remove the hemoglobin from the red blood cells after he exposed the ghost to very low oxygen concentration. They still retained their normal shape and showed no signs of sickling. Why is that important? Hey, in the back, why are we still turned side? Oh, we're not. My bad. I hear talking instead of listening to the question. So we have a ghost. We created these ghosts that we removed the hemoglobin and we put them in low oxygen concentrations, and still the cell was actually normal. It did not sickle. The cell with hemoglobin, by the way, he called those ghosts, correct? The cell with hemoglobin when put in low oxygen concentration, what did it do?
It's sickle.
It's sickled. Why is that? Why did that test show? Yeah.
The correlation of hemoglobin levels to sickling.
Yeah. The fact is the hemoglobin in low concentrations of oxygen is what's sickling the cell. Right? Why did he have to do that? Because it could have been some other component. We know now that what causes the cell to sickle, it's the hemoglobin in people with sickle cell disease causes the cell to sickle in low oxygen concentrations. By the way, that means the brain, the heart, the lungs, or not the lungs, the kidneys, anywhere away from the lung. Yeah.
Do they have a different hemoglobin concentration than like people without sickle cell anemia?
My understanding is no. The concentration of hemoglobin is the same. The difference is in the quality of the hemoglobin, not the quantity. It's a good question. Now they went and started talking about water and concentration of water and looking at concentrations, which I think everybody should be comfortable with, correct? And we know a 500 millimolar. You may not remember what molar is, but it's a mole is 6.02 times 10 to 23rd of any particle. So a millimolar is a thousandth of that. It's just a measurement of concentration. That's all you have to know, is it's a measurement of concentration. So how much of something? So 500 is more than 300, okay? Which is more than 100. Are we okay with that? Yeah. As long as the units are the same. Always look for the units. Even if you don't know what the units are, you know 500 is more than 100. Correct? All right. As long as the units are the same. So, go ahead. Saline is a standard. Saline means it's the same salt concentration as your blood. So it is salt. It doesn't mean salt. But when someone says saline solution, they mean a solution that's the same. It's hypotonic to your, isotonic to your blood. All right, guys, we'll finish this on Monday. I'll post the answers. I will post the answers, and you can finish it on Monday if you'd like.